This is episode 276 of the Stem Cell Podcast, Musculoskeletal Disorders with Dr. Dmitriy Sheyn. Hello, everybody. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Dmitriy Sheyn. He's from Cedars-Sinai on the podcast to talk about his research on the development of stem cell therapies for musculoskeletal disorders. We've also got our usual roundup of recent highlights and stem cell news coming up.
But first, before we get to my neighbor, Dima, we'd like to introduce Muscle Cell News, a free weekly newsletter provided by Stem Cell Science News. Muscle Cell News summarizes all of the latest research, news, jobs, and events in muscle cell research and delivers it right to your inbox every other Monday. Save time and keep current with Muscle Cell News. You can subscribe for free at www.musclecellnews.com. We're going to start things off with something that's not a stem cell story, but it's pretty fun, and maybe it has some applications for in vivo modeling or studying stem cell transplantation in vivo or something like that. It's a fun article. It's a tool that's sort of like obvious in some ways. That's actually gotten a lot of popular press attention. It's a science paper titled Achieving Optical Transparency in Live Animals with Absorbing Molecules. And if you're on social media, you probably have seen some of the videos and some of the images from this pretty neat study. It's actually coming from Gosung Hong and Zihao Ao. Gosung Hong is, I believe, at Stanford, and I think this is a paper that was put together by Zihao Ao when he was a postdoc, I think, in the Hong lab at Stanford. They do some cool stuff in the Department of Materials Science and Engineering. I've had some collaborations with, not them, with some other folks in that department in the past. And I think now Dr. Ao is at UT Southwestern, I believe. Anyways, it's trying to solve an issue that has been around for a long time. And that's, how can you best image the anatomy of live animals, right? You know, there's different ways you can do this, of course. There's ultrasound, there's MRI, there's all different like bioluminescent modalities that have pros and cons, but the dream is to take that sci-fi approach and just like put on a pair of glasses or something and be able to see through the skin and look at the innards as they're actually doing your thing, right? Seeing the gut move around, seeing the heartbeat just in real time as the animal is alive. And the dream is ultimately that something like this could be applied to humans, right? So they had a really fun trick that they thought of here. They used something that's a very common food coloring approved by the U.S. Food and Drug Administration. This is tartrazine. And funny side note, this is in all different kinds of food. Most famously, this is that orangish dye that gives Doritos their quintessential color, that, you know, bright, fluorescent, toxic orange that we're all familiar with when it comes to eating Doritos, right? Apparently, that Doritos food coloring has a really interesting optical property where, and I don't know the details of the optics behind this, but they did something called, they applied this thing called the Lorentz oscillator model. I'm not going to go super deep into this. Ultimately predicting that certain dye molecules with really sharp absorbance resonances in near ultraviolet spectrum, which is 300 to 400 nanometers, and the blue region of the visual spectrum, which is 400 to 500 nanometers, are effective in raising the real part of the refractive index of aqueous medium in longer wavelengths when dissolved in water. Okay, that's a lot. That's a lot of optical physics first thing in the morning. But long story short, if you can dissolve some of these dyes, such as this tartrazine, in water, it can reduce the contrast between water and biolipids, of course, in cell membranes, which is ultimately leading to optical transparency of live biological tissues. So, to simplify it for us dumb stem cell biologists, I suppose, they just dissolve this tartrazine in water, rubbed it onto the mouse skin, waited a couple of minutes, and then we're able to basically look through the skin itself when you shine the right light wavelength on it. It's pretty wild. And I can just talk about this, or you could just go, look at the images and videos on social. But yeah, they conducted these experiments both in hydrogels, like tissue mimicking hydrogels. I think they use like a piece of chicken or something and just like rub some of this tartrazine dye and dissolved in water on that chicken and saw it became transparent. And then they did it in the mice as well. So they transformed the typically opaque abdomen of a live mouse into a transparent medium. And this transparent abdomen allowed for direct visualization of a bunch of different things like fluorescent protein labeled enteric neurons, capturing the movement of these enteric neurons that mirror the gut motility in live mice. So they're looking at gut movement, gut function in live mice. They also looked at cerebral blood vessels as well, again in real time. And they also looked at the mouse hind limb for high resolution microscopic imaging of individual muscle sarcomeres. That's pretty incredible. And yeah, that's basically the story. It's just a tech development story, sort of like a low hanging fruit, where this very commonly used dye, if used in the right way, can lead to some beautiful, optimized imaging of live animals. And of course, the dreams, like I said, to apply this to humans. And the big limitation here is that mouse skin is way, way, way thinner than human skin. It's like 10 times thinner than the skin that we have, right? And so instead of like just rubbing this on human skin, maybe you would have to inject it, something like that. But I'm sure they're looking into it because everybody's going to be asking this question, especially for clinical applications, right? Do we want to have something that's perhaps even better than MRI or ultrasound for live imaging that could ultimately be used by clinicians? So, pretty cool story. I'm excited to see where this goes in the next couple of years. And maybe we'll use it in our own lab too.
I got one word to sum up this story, and you said it. You said it early on, innards. And you describe it as the dream, and I'm kind of like it's the nightmare in some scenarios. I don't know. It's absolutely the dream and the application, but like looking, you guys got to look at this story because it's really striking how this relatively, I guess, I would call it elegant, right? Outside the box approach it let you look at these pictures and you see just all the gut squirming around there. It's kind of gross. But the resolution is super high to the point where I kind of got like a Blair Witch thing going on with the camera shake on there, visualizing something in real time. And I was like, I mean, my eyes were kind of shaking around. But that I think underscores how like looking at a live system really gives this whole other dimension. So an amazing application. I just gotta say about this was like everything that comes out of Stanford. I know you're Stanford alum, fanboy, and all that. No fault. It's a great place. But like, do they really have to like slap you in the face with the Stanford thing? And to the point of in this case, like half the figures, the way they illustrate the optical clarification is by taking like whatever, like you said, chicken skin or something. They put it over the Stanford logo and you can't see it. And then they clarify it, and there's a glorious Stanford logo. I mean, get over yourself, Stanford. But clearly some kind of like patent background there. They're smarter than I am, and that's like a little bit of branding that they slipped in the story. So what good show, mates. Congratulations. And last thing I'll say is that yeah, like in terms of application, it really is amazing, and I love that outside the box approach, like as I referred to there with like that thing that's present and out there already approved for use, and lo and behold, in the Dorito dust. Hello. I mean, maybe I know the skin is thicker for us humans, but maybe if you add enough of that Dorito dust, we can get it done. And for me, that's a bonus because that stuff is like crack.
It makes me wonder what Doritos are doing to the inside of your body, right? I don't know. It's gonna keep me up at night. But you're right, Stanford loves their branding. This is not the first time I've seen something like this. I've seen a lot of papers where they work in the Stanford logo somewhere in some figure. But shout out to these folks. It's a really great technology. Can't wait to see it come to humans eventually and hopefully the next couple of years.
Let's see. Let's see. Dorito lining up. The other one's going to make a lot of money out of this. Frito-Lay and Stanford. Moving on to a story. It's another adult organs, not as gross. No innards here. But very important, highly relevant story about muscle, right? Beach season is over. We're all getting flabby. But this is really, I mean, in all seriousness, talk about muscle wasting diseases and injuries, a major unmet need. And muscles are renewed, regenerated by satellite cells, which are the muscle stem cell. And they mediate muscle growth and regeneration. Um, and they do it in this niche, right? Where they lie quiescent until they're activated in response to injury, divide asymmetrically, self-renewing on the one hand, and also forming these proliferative progenitor populations called myoblasts, right? Which those further differentiate, fuse to form myotubes, which are the functional component of skeletal muscle. And then the other half of that self-renewal event, the satellite cell, can then return to quiescence, go back to the niche, repopulate that niche, and chill until the called into action the next time. Of course, this is, I would say prime candidate, maybe number one candidate, an adult stem cell like blood, for example, that lies in a niche and quiescent and can activate itself renew. I mean, it's just like the blood. Why aren't we applying muscle stem cells, satellite cells in therapy? Well, despite many attempts, not a new idea. I didn't just come up with it, brilliant as it is. Um, Despite decades that we've been trying to do that, it's not really understood. How can you expand these satellite cells in culture while maintaining their stemness? So you put them in a conventional culture and they go to this myoblast progenitor fate, which is capable of producing these muscle fibers once you transplant them, but like at the scale of 1,000-fold worse than the satellite cells. And those satellite cells, by contrast, they can, as I said, repopulate the niche, they self-renew, and they can regenerate muscle like over and over and over throughout the entire life of an organism, presumably in humans too, but in mice, yeah, forever. Till that mouse dies, you got these muscle cells working for you. So the question is again, how do we get these cells to expand in culture? How do we make them like a tractable, renewable asset that then we can deploy in these therapeutic contexts? Well, Lee Rubin at Harvard has been working on this for some time, and in Nature Biotech, he has a new story uh, where he explains pretty much and sets out this method for expanding satellite cells in vitro, and it's really about organoid. Like it's surprising to me that other people have not uh, shared these data already, given how deep the stable of high profile muscle stem cell researchers there are. But going into 3D, making an organoid out of primary muscle tissue was, and of course, some refined conditions. I don't want to oversimplify. But they were able to get these in vitro derived satellite cells, as they call them, with these skeletal muscle organoids, and showed that they could transplant them like with really small numbers, showed phenotypically that they were the true product, and transplant them. They could turn into myofibers and repopulate the niche, also generating muscle in multiple rounds of muscle regeneration. You got to look at these pictures. It's like the whole cross section of these muscles are from donor cells. And improve force production. I think a key metric here is showing that they're actually capable of conducting force and improving function. They show also in this model of muscle wasting, They show that they can recover and repopulate in that context also. They did a bunch of, omic analysis to show that these in vitro derived cells uh, were true to the satellite cell. And then finally, although this was not as well fleshed out, they show that they can generate these cells from human muscle biopsies as well. Not necessarily generate them in a tractable way. I would say the one shortfall there is they show they could get conditions where they expand and they look in the organoids and find that those satellite cells are the one thing they fell short of there was being able to purify them. Didn't have the surface markers to purify them and show like the more functional repopulation studies that you would want to see. But for me, I think this is a really big idea that is not a new idea, right? But I think it's the culmination of many years of work from many groups and the application of a recipe and approach to generating cells that like in a really robust, scalable way that I think is going to be clearly here with the last experiment with the human cells is kind of the preclinical study to set up for trying to look into these cells actually in I guess large animal trials first, but finding a human correlate and seeing how well they work, which could be a game changer for a huge swath of unmet need related to muscle wasting and injury.
Yeah, and I think that's what you're saying there at the end. I think that's exactly why this is a nature biotech study, right? Because nature biotech is all about that translational element. I mean, there's another story that you might talk about in a little bit that has this similar vein. But you're right. I mean, ultimately, it's about the human, right? Can we do something like this comparable technology, comparable technique in a human system, in the human setting? And the the human data was sort of slapped together at the very end there. It's a supplemental figure, and it's almost seems like to me that was done in response to a reviewer comment, which understandable. That's probably how it got to the level of a nature biotech. But that's ultimately what they're going to be looking at here in the next couple of years. Functionality, like you alluded to, is a big deal here as well. I'm glad they're able to flesh that out. As the dumb IPS biologist in the room, I'm wondering if there are some parallels here for iPS derived satellite cells as well. I know, if you have the primary and you're able to expand that, then why bother with the IPS? And they actually mentioned that in their, I think, intro. They're like, yeah, IPS, we could do that too, but they're of course immature. They have all the issues with IPS that we're aware of, but maybe they could do something comparable with IPS derived as well and create some organoids in that way. But yeah, I think a lot of really promising stuff that's going to come out of this paper, and that's sort of what nature biotech is all about, right? That translational element.
Absolutely. And you touched on so many important things there, which I just want to briefly talk about. One, yeah, I mean, you mentioned it, IPS derived correlate would be, I think, critical because one thing you got to consider is that, especially in the context of genetic disease or even muscle wasting with age, right? What are those primary cells going to look like? Are they going to be capable of the same regenerative capacity? Are they going to be capable of like gene correction in order to address disease. So yeah, we really got to do a lot of human studies to see what the longevity of those cells are outside of a system like mouse. And you address that final experiment in the human, and I think it's an important footnote here that yeah, that was certainly a response to review. And Nature Biotech, as much as it is at the forefront, like the reviews for Nature Biotech are exhaustive and extensive, but also I would argue too long. This went into review or was submitted January 2021. We're approaching 4 years that this paper was in review, which in my position, I don't know. I guess the data wasn't there, but I'd be willing to bet that it was a few small things that held this up as opposed to the core idea. The core idea here is so straightforward. So maybe we got to do better as reviewers to try and elevate things that are timely and important and need to be built upon in a shorter term and shorter time scale than 4 years. I mean, come on. And lastly, though, in contrast, I will say to echo your point, Nature Biotechnology is a forum that addresses, and introduces tools that are going to be applied widely. And this is another example of that. And I referenced the core, the the kind of kinship, I guess, between muscle stem cell and hematopoietic stem cell, and the curiosity surrounding why muscle stem cells haven't been applied in the same way as hematopoietic stem cells. And this is an interesting opportunity, or appropriate opportunity here, to introduce like a little kind of addendum in the roundup here to another big story that came up in Nature Biotech a couple weeks ago, which was, at long last, although there have been multiple stories making this claim, the generation of bona fide hematopoietic stem cells, engraftable hematopoietic stem-like cells, I guess you call them, from iPS cells from the Stanley lab. And this is a big story. I mean, huge. We've talked about it as the Holy Grail. I think we got to mention it here because we didn't feature it in the roundup, but it's an important story that I think you guys should take a look at, because it sets out some pretty impressive data. There are going to be a lot of haters out there, because, yeah, there's no like fundamentally different approach in terms of how mechanistically it's 34 positive cells, a very clean diff. But I think there's going to be a lot of studies forthcoming to see how real those cells are. And it's a nice companion point to these muscle stem cells, true hematopoietic iPS-derived cell. I mean, this is truly a wonderful time to be alive. I can't wait to see how that experimental landscape plays out, Arun.
Yeah, that was a big time paper that came out, and I'm glad we highlighted it in some capacity. I was having a few chats with folks in our department who are hoping to take that protocol and just run with it right off the bat. There's folks here who, like, at Cedars, who want to bring that protocol in house, and why not? It's a pretty straightforward recipe. It's a diff, right? There's nothing, as far as I know, there's nothing genetic about it. There's no genetic overexpression or anything. It's just a combinator. It's just a cocktail. So theoretically, anybody might be able to give it a shot. Of course, and you would know about this more than me. I think one of the huge drawbacks of that study, or weaknesses, is the secondary transplantation aspect of it. I was talking to some HSC biologists about that. So the one thing you want to show is that the cells that you're making, the iPS derived HSCs, can be you know grown indefinitely, right? So if you take the cells that were transplanted into a mouse or whatever model system, uh, can you take those same cells after X amount of time and put them into another mouse and have that indefinite secondary growth? I think the data on that side of the paper was a little bit weak. I think the N values there were pretty small, but the primary part of it seemed really good, in terms of just making these cells from the iPSCs using the cocktail. So inevitably, this is one of those things, sort of like the derivation of iPSCs themselves, that like everybody has their doubts about initially. But the hope is that once enough labs start using it in their labs, they'll be able to see, wait, this actually works. I really hope that that's the case, because, like you have alluded to many times here on the show, this is a holy grail. This is a game changer. So we really want to we want to make it work. So moving on to the third story of the roundup. This is something in my neck of the woods. This is a cardiac story. It's a cardiac assembloid story, which is kind of fun. You don't see a ton of those. Assembloids are more often in the realm of neuro. You're talking about different lobes of the brain, different parts of the brain that are kind of shoved together in these organoid models, right? But the heart has different parts of it as well. You got the atria, you got the ventricles, you got the nodal conduction network, and that's exactly what they're trying to approximate in this cardiac assembloid story, coming from the lab of Harsha Devala over there in Amsterdam, the University of Amsterdam, the Amsterdam Medical Center. First author here is Jiru Lee. It's a cell stem cell paper titled "Modeling the Atrioventricular Conduction Axis Using Human Pluripotent Stem Cell Derived Cardiac Assembloids." So, um, there's all different aspects of the heart. Like I mentioned, there's different parts of the heart. It's not just a single uh, homogeneous chunk of muscle. That's not true. Of course, you have the conduction network that's really critical to provide the electrical continuity between different parts of the heart, like the atrial ventricular chambers. There's this atrioventricular conduction axis, right? And so, as a cardiac biologist, and as cardiac biologists know, there's typically assumed to be three different classes of cardiomyocytes. One, atrial cardiomyocytes found in the atria. two ventricular cardiomyocytes found in the ventricles, which are the ones that perhaps pump the hardest, and then three, a really unique population known as nodal cardiomyocytes, which are involved in the conduction network and making sure the proper action potential is flowing across the different chambers of the heart, right? So the nodal cardiomyocytes that populate the atrioventricular conduction network or AV canal in the embryo or AV node from the fetal stages onward, they propagate impulses slowly. Okay, they're different from the other two types of cardiomyocytes in that way, and they have a slow propagation of the action potential. This is really important because it's ensuring sequential contraction of the chambers. You don't want the atria and the ventricles to contract at the same time. You want the atria to typically go first and have a slight delay, and then the ventricles after that, right? So, but there's a lot of diseases in this where the situation goes wrong, right? There's dysfunction of the AV nodal tissue, which can cause really severe disturbances in rhythm and contraction. And as we know, arrhythmias can be fatal. So this is a major area of interest in terms of clinical applications, but also just basic science, studying what goes wrong in these systems. And so you need human models that can capture some of the features of the AV node. And those are pretty limited. Even some of the mouse models are pretty limited as well. So here they're reporting an approach for the reproducible generation of AV canal cardiomyocytes, which is in itself a big deal. But then they took it further and then applied those AV canal cardiomyocytes to an organoid model. Now, they did the standard characterization. They compared the iPS-derived AV canal cardiomyocytes with the primaries, right? The real deal, as you have to anytime you're trying to validate a cell type created from a diff. So you compare the gene expression, and they showed that they have in vivo-like gene expression, in vivo-like electrophysiological profiles. I don't think anybody in the cardiac field is going to say right now that the cardiomyocytes you're making from IPS are one-to-one perfect replica of what you find in the adult human heart. It's just not true. There's so many electrophysiological differences and even gene expression differences, but it's a model, right? So here they created those AV canal cardiomyocytes and then took them further and built them into assembloids comprised of atrial, AV canal cardiomyocytes, and ventricular organoids. So those three different types of organoids, smash them together to create an assembloid. And the cool part about this actually recapitulated that uh, conduction system, the AV canal conduction system that I was alluding to in the beginning, where these nodal cardiomyocytes have a slower conduction, right, in comparison to the atrial and ventricular. So you have that fast, slow, fast activation pattern that's typical for the vertebrate heart. That's really neat that you can replicate that even in this artificial system. And then they applied it, right? They utilized systems to reveal intracellular calcium mishandling as the basis of laminin A associated AV conduction block, which is a genetic condition, and ultimately just a really cool, really useful system to show one, a novel cell differentiation in the AV canal cardiomyocytes derived from pluripotent stem cells, but then two, a neat tissue construction and or assembloid strategy to help model these complex disorders that affect heart rhythm. And like I mentioned, this is a big deal because these diseases are often fatal. I mean, you've heard instances of athletes, for example, succumbing to cardiac arrhythmia. And it's a really scary thing at times. And most recently, there was that example of Damar Hamlin in the NFL, right, who was took a wrong hit to the heart and caused some sort of arrhythmia, and passed out on the field and was very close to death. So understanding how the conduction system works, I think, has a lot of very important applications.
Yeah, and that for me, the most impressive part of this, which I know that's the whole point, I guess, but how they were able to, recapitulate that the directionality of it by the differential, ordering organization of those organoid components of that assembly. I thought it was really, again, elegant. Like it seems almost, I guess, relative to where we will be, and I that's the vision I have. But relative to the vision I have, it seems almost kind of basic and rudimentary. You make these arrays and then you just smush them together. Like the fact that you're able to glean some insight and recapitulate biology and force, right? The biomechanics there, I think, is really a key advance here, and I think presages an era that we're moving into now. I think we're right on the cusp where it's like more of this bioprinting and this ordered uh, differentiation and assembly. Right, we saw a bunch of stories which we talked about on the show at the ISSCR, like a la Lutoff and others, neural tube printing, etc. That we talked about the somatoid printing, but I think now the idea of putting these neural assemblies of organoids in a ordered kind of cast or mold type format, I think is going to be really exciting, and I'm looking forward to those studies.
Yeah, and I think it can potentially improve the homogeneity of these organoids, right? Because that's a limitation. They actually mentioned that directly in their limitation section. And we all know about it, right? Like organoids have heterogeneity in their production. I think a lot of people are trying to get around that. The best approaches are able to create really homogeneous organoids. But if you're taking that one step above and turning these into assembloids, that homogeneity is going to be even farther away from the reality. So they mentioned exactly that. They think like custom plates or inserts or even 3D bioprinting could be used for future studies to actually improve the success rate of making these assembloids and again making them more homogeneous. So I mean that's ultimately very important for modeling, drug screening, whatever application.
Yeah, amongst those applications is trying to model neurodegenerative disease, right? That's my segue, or the best I could do. I got a story about like more generic uh, or degeneration and regenerative approaches to that degeneration. Not the best toss, but I'm trying here, Arun. This is not a brand new idea. In fact, it's kind of an old idea, and we've covered it. It's this idea of cellular reprogramming using the Yamanaka factors, OSKM, enforced expression in like an old mouse to rejuvenate it. And this is, I think, a more refined application of that. And more specifically, it's in science translational medicine, which I think is says something about where this story is and where the application is envisioned and how close we are to maybe moving in that direction with real, human clinical trials. And of course, it's about senescence, cellular senescence, right? Which is, it biologically exists as a tumor suppression mechanism, right? You don't want cancer, and it's implicit with aging, right? But that cellular senescence comes with the expression of a lot of like cytokines and even growth factors that are kind of maladaptive, collectively known as the senescence associated secretory phenotype, which can have a deleterious effect on tissue microenvironment, uh, and can ultimately, really, I think, at the business end, leads to dysfunction, a reduced competence of stem cells. So tissue renewal, the tissue itself, the microenvironment is undermined, as well as the cells that have potential to repair, right? So there were all these there were multiple ideas and approaches to senescent cell clearance, driven in large part by these ink 4A, I think, P16 ink driven constructs, uniquely active in senescent cells. The idea was you could have these like senolytics, right, where the senescent cells would die off, and it worked. You get improved health span. That's the key here, not just lifespan, improved health span in mice. But I didn't really know this actually. But I think it's an important point that senescent cells, and specifically like the senescent cell niches, can be really important for normal physiology in multiple contexts, including the lung and the liver, where there's this quiescent senescent cell niche that provides essential instructive factors, and clearance of which can have like a harmful consequence, right? So, senescent cells, it's a problem if you just wipe them all out, right? So enter the Yamanaka factors, right? And this is a idea introduced by the king, El Rey, Juan Carlos Ispizua Belmonte. Here now, another story follow up from that. He's at Altos Labs. But the original approach by his and other groups was expressing the OSKM, the Yamanaka factors, OCT, SOX, KLF, and MIC. But that, it was an issue, obvious reason, safety, really. Especially when you have like an unregulated expression, mostly the MIC. MIC's not good. Even so, there was like this short-term short-term cyclic expression of OSKM in model of progeria, as well as in wild type mice that showed improvement in health and lifespan. But the problem here is that it wasn't cell type specific, right? And you can see where I'm going here. You would have short-term and cyclic expression of these factors, but it wouldn't be in a unique subset of cells. It'd be in all cells, not all, but not discriminating. And that led to some, well, potentially a lot of risk there in the healthy cells. If you're gonna blast the Yamanaka factors there in healthy cells, you don't really want that, right? So enter here a more refined approach, right? And this was a adeno based. So again, this kind of transient, non-integrative, single dose of adeno associated viruses that had OSK, not MIC, just OSK. And here, this is a key under control of the CDKN2A promoter, right? And this gene is primarily or typically active or upregulated in aged and stressed cells, right? So the idea there is instead of having blanket expression, they target it to these senescent cells, which they could use a little shot of OSK, right? They showed in the same progeria model that the mice, their lifespan was extended. They showed a rescue of multiple niches, specifically like reduced pro-inflammatory cytokines, and in the bone marrow and spleen, the hematopoietic complement was kind of rejuvenated, which is not exactly surprising in terms of that compartment being affected, because it was introduced by tail vein, which is going to, of course, pile up in the liver and going to be disseminated throughout the vasculature, including the marrow, right? So not surprising there, but I think the extension of that was also impressive. They took just wild type mice that were OAF, as we like to call them, these aged mice, and gave them an injection and showed that you could extend the lifespan. And just to put it into like human-centric terms, they went from like 800-ish days median to 900 days. I think loosely approximating, you say, hey, extend human lifespan, maybe 80 to 90. I would take that, especially if it was a solid health span where, playing some tennis or some pickleball at least. That was a big hit. And also here, another, I think, really a la the science translational medicine positioning here, they had this nice experiment where they showed accelerated wound healing. So this wasn't systemically delivered. They injected the AAV directly into the skin and wounded skin and showed that they could get, healing in aged mice, which was presumably compromised in aged mice. Normally, they could get the accelerated wound healing, and then they patch on, as same way they did in the other story I just talked about from Lee Rubin Lab. They showed in human primary fibroblasts, they took primary lung fibroblast cells and showed that they could kind of rescue this inflammation related phenotype using this approach. So this, I think, was again, in that, I think that this is really being positioned here for clinical trial, showing not just like a more refined approach here, but showing, I think, a modality in terms of the skin, which is maybe a middle ground, wound healing, before you're injecting this systemically into the marrow, right? Which I think is also a good endpoint. I'd be down for that, particularly if I had progeria. But I think that skin injection may be a middle ground here where we might see this, which I would have never predicted. I thought, Yamanaka normal biology, no thanks. But now I don't know. Now I don't know. I think particularly for like an elderly cohort here, where there's not a lot of options, maybe this OSK approach could be a second life.
Yeah, I'm with you. I like when I first heard about this, and the work that Altos Labs is doing when it comes to adding the Yamanaka factors for clinical therapy, I was like, no way. It's because the first thing you think of is cancer. But hey, that's what they're addressing here, right? Like what you alluded to at the very beginning. MYC is that prototypical oncogene, right? And if you maybe take that out of the fray, things maybe get a little bit safer. So here you're just talking about OSK and more targeted delivery. But hey, here we are, 2024, and we have Altos Labs papers coming out in Science Translational Medicine. How about that? Shout out to Juan Carlos Belmonte, who's really a pioneer in this field, as well as all the folks who have gone from his lab over to Altos Labs, which has a funding that's bigger than some small countries. I got to say, very well funded biotech startup there, but understandably so. I mean, they're working on a very hot topic, a universally attractive topic, I suppose, in reversing aging. Everybody's looking for that fountain of youth that we've alluded to before on the show. And I think this is a really cool paper that's, like a case example of how this might work in a mouse model. So um, I'm still obviously very like I wouldn't want to receive these personally, like right now. If I had progeria, maybe it's a different story, right? My fear though is my fear in this area is that there's going to be some of those biohacker types who are going to go ahead and take some of these OSK AAVs and just start injecting them into their own bodies and are going to get cancer or something negative is going to happen, and then maybe it's going to shut down the whole field, because the concept here is sort of simple. You just get the Yamanaka factors and you inject, and hopefully it's not that low hanging of a fruit, right?
Yeah, I never thought of that, but you're right. The barrier to entry is very, very low. I don't know if this stuff's on Agene, right? But if it were, it'd be pretty easy to get out there, make some adeno, and reset the biological clock, so to speak. I wouldn't mind if you got some adventurous biohack. I'm not advocating for this, and let me say here that the opinions expressed by myself and Arun are not necessarily shared by STEMCELL Technologies. But if you biohackers want to get out there and give it a shot, I'll give you my email. Let me know how that goes. But in all seriousness, no. All credit to Juan Carlos and his team, who have not only the resources but the endurance to take what I thought was a bit of a far out idea and bring it close to fruition. And while you were skeptical, maybe I'd say is a fair position, give it a few years, partner. When you got your sciatic nerve squawking at you every morning, maybe you'd be in for a bit of OSK AAV. And, maybe we could talk to Dima a little bit about that. An alternative to his approach for my aching back, maybe some OSK, although for now I'm going with Dima. We can talk about that at length with him in just a minute. But before we get there, I got a quick message from STEMCELL Technologies, which is on point today, because if you don't already work with skeletal muscle progenitor cells, after the stories we have told you today, namely that from the Rubin lab, maybe you got to look into it. The STEMCELL Technologies human myocult workflow supports your muscles research from start to finish, allowing you to derive, expand, and differentiate human skeletal muscle progenitors. You can also expand mouse myogenic progenitors using the mouse myocult expansion medium. Learn more at www.stemcell.com/myocult. All right, you guys, on today's episode, we have one of Arun's homeboys from Cedars-Sinai Medical Center. He's associate professor in the Department of Orthopedics and in the Regenerative Medicine Institute, Dr. Dmitriy Sheyn. The Sheyn Lab focuses on the development of stem cell therapies for multiple musculoskeletal disorders caused by acute injuries and aging. Using gene editing, biomechanical forces, and specific transcription factors, the Sheyn Lab seeks to direct stem cells towards skeletal tissue-specific cell types to support healing and to reverse degenerative and inflammatory processes. Dmitry, thank you so much for joining us on the show today.
My pleasure.
Yeah, special welcome to my next door neighbor here at Cedars, like you alluded to, Daylon. Welcome, Dima. It's good to have you here on the show. You're actually recording this about 2 offices down, so this is local, I guess you could say. Um, I know you've had like this really productive last couple of years here at the RMI at Cedars, securing some big time CERM, NIH grants, and publishing some really cool papers in the orthopedics field and the regenerative medicine field. Before we dive into the details of the papers, why don't we just start off by giving us an overview of kind of what your lab is focusing on right now, just in your own words?
Right. So, yeah, as Taylor mentioned, we working on few orthopedic disorders or conditions, and the common denominator is using stem cells and mainly iPS-derived technologies to address those disorders. So, orthopedics is very broad or prominent in the population, and the numbers of patients are enormous. So, but we don't have good biological solutions. We have a lot of hardware solutions and treatment that are quasi biological, but not proper stem cell regenerative solutions. And that's what we think that we aiming to do. And our main focus is spine research and low back pain. That's kind of half or more than half of the lab is working on. But we also fascinated by tendon and ligaments. This field is relatively young in the stem cell context. And another thing that we're working on is osteoarthritis. So specifically post-traumatic osteoarthritis. That field is not young, but there is not much known on stem cell context, and we're trying to kind of interject a stem cell solution where other solutions have failed over decades that has been studied.
Yeah, as you said there, the scope of musculoskeletal disorders is very broad and diverse, right? And I think almost anybody can identify with that, the actual firsthand, either themselves or a family member. I can speak for myself. I've struggled with lower back pain, and it's so amorphous. It's and so frustrating, right? Because it doesn't feel like there's any solution but to just wait. Much less, the idea of mitigating the repeated episodes of lower, in my case, like it's something that's never going to go away for me. I didn't think it was until I looked into your research, and that's part of the problem, right? I mean, in aggregate, these musculoskeletal disorders, they constitute this massive healthcare burden, which is like an everyday burden, particularly in the developed world. When I think of stem cells and musculoskeletal diseases, as you said, there's not a lot of these regenerative solutions. But when I think of like the scope of disease that could be addressed with cell-based therapies, I'm thinking like muscular dystrophy or sarcopenia. But the scope of musculoskeletal diseases writ large is much broader, much deeper. I looked into some of the stats. I mean, I don't know how on target these are because I don't know how you would get the exact numbers, but healthcare spending, from what I saw in musculoskeletal disorders in the US alone, was approaching $400 billion when it was last counted. And that's not counting the substantial loss of productivity among those affected and the resultant blow to the economy. I know when my back goes out, I'm out. I'm on my back for a week. So the unmet need here is massive and looks to be increasing even more, right? Because increased BMI, which is a major contributor to the prevalence of musculoskeletal disorders, showing no signs of abating in post-industrial economies. So I feel like this problem is expanding, is looking to expand. But the research funding on the flip side is relatively scarce. I looked at the study; it's about 10 years old now, but it estimated that the NIH allocated $4 of research funding for every U.S. person affected by chronic pain, and musculoskeletal derived. That's compared to 40-ish dollars for diabetes and like $400 for cancer. So 2 orders of magnitude there span. And of course, I mean, this is numbers, and maybe it's because like literally everybody has chronic pain relative to cancer, which is relatively less prevalent. But I would ask you, what do you think accounts for this funding gap, given the scope of the problem? Is it that chronic pain isn't life or death? Is it that so many other, as you said, like hardware solutions or pharmacologic solutions are already out there? What are your thoughts there?
So thank you for those stats. It's really important to note. And yes, it's definitely very underfunded. And I agree to both reasons. First of all, yes, it's not life or death, right? So and yes, if there was recently studies saying that you can just walk it off, right? So walking decreasing low back pain. So these things happen, but uh, besides this huge scope, we really don't know enough. Like we don't know when it's cancer. We know all the genes that cause this. When it's a genetic disease, there's one mutation that we know what's causing it, right? Here, because of the prevalence in the population, because of the diversity of causes, and specifically in back pain, if there is an injury, okay, we know this is what the initiation of the process of the problem is. In back pain, you have pain. There are tens, if not hundreds, of reasons that can cause it. And even today, when you go to spine surgeons, they don't know specifically which level of intervertebral disc causing the pain. So we so in the dark in that perspective, and pharmacological solutions, help some surgical intervention work, right? So if you completely lost your discs and you in huge pain, you go to spine surgery, they fuse with all this massive hardware, they fuse your spine. You can after a certain time walk again, right? So it's some of those problems are solvable, so to speak, but it's never a good solution. It's never. There are so many risks in massive surgeries. There's so many. When you fix one level, it will come again in another level. So there are solutions. They're not biological mostly, and that's why we think that biologically relevant stem cell therapies will make the difference, right? But yeah, you're right. The NIAMS, the NIH Institute that funds musculoskeletal research, is manifold smaller than cancer or any major institute. Although it covers yes 80% of the population.
Hmm.
Yeah, so I mean, it's a huge unmet medical need affecting almost everybody. We all know people who have back pain, right? Like I'm starting to get back pain myself. I'm not that old. I'm not as old as you, Dylan, but I'm starting to feel it. I'm starting to feel it a little bit. But I think you've established a really important niche, right? You've established this great niche and potentially, like what you're alluding to, using stem cells as a therapeutic option, biologics as a therapeutic option. I mean, one thing that you're hoping to do is, generate a bunch of different cell types that are related to different orthopedics treatments, such as treatments for back pain from human iPSCs, like you're alluding to. I mean, this isn't directly related to back pain, but an example of some of the stuff that you're working on are generating tenocytes, for example, the specialized cells of the tendon and the ligaments that secrete and build extracellular matrix. I mean, you actually had this really cool bioarchive paper that you posted recently that was using single cell transcriptomics to figure out how to drive iPSCs to the tendogenic lineage. And I'm actually kind of shocked this hasn't been done before. And maybe it's thanks to single cell that you can figure out the developmental timing and developmental signaling that you actually need to make tenocytes in vitro. So tell us a little bit more about this work generating tenocytes and maybe its implications for other types of cell therapy.
So, as I mentioned, tendon is relatively young field in regenerative medicine. So the first tendon conference I attended, I was amazed how there are two a very distinct population of scientists in that meeting that probably never talk to each other. Pure developmental biologists, right? That they know where the cells are coming from, all this lineage tracing, mouse studies, zebrafish, Drosophila, and clinicians that just want to fix things and they don't have anything in between. So I think, and that was how it was for decades. And there are not good models for some of the really important and prevalent tendon disorders like rotator cuff, right? Rotator cuff is the main tendon injury, and people cannot lift their cup after that, right? So that's really a problem of in thesis, right? The connection of tendon and ligaments to the bone, and we don't have that, research enough at this point. But it's coming up. I think people realizing that this is a niche or field that was not explored, and we're excited to contribute to that research and develop these therapies. So it's also very challenging because tendon has very specific function. It has to have this very unique biomechanical properties. It has to be strong but elastic, right? And it has to transfer the force from the muscle to the bone without being too stiff, right? Or too elastic. If it's too soft, then muscle will work for nothing without moving the bone, right? So these properties and achieved by certain matrix, certain patterns, and certain structures that are not easily, replicated. So there is a huge challenge in tendon regeneration. Right now, what we do is allografts, autografts. You take a tendon from another place and put it where it's missing. There is a lot of clinical success with it, but that's not ideal. There's a comorbidity or side comorbidity with those surgeries. Allografts are not really integrating; they fail eventually. So we're trying to replicate tissue that is really difficult to replicate. And for example, bone is much easier, right? It has very specific factors that if you bombard the tissue with the BMPs, there will be bone. It has a paracrine effect that it kind of self-healing. I mean, we all know that breaking bones is not the end of the world, right? If you're relatively young and if you didn't crush, if it's not a huge fracture, then it heals pretty fast, right? Tendon is not like that. Tendon rarely heals, and if it heals, it's usually heals with scar tissue, which doesn't have the same mechanical properties as tendon. So yes, you can bridge the gap with scar, you can kind of walk, but it never fully regenerates, and that's where adding stem cells with probably biomaterials that are appropriate for that tissue may help.
God, I hope so. I mean, I'm not just an old geezer when it comes to my aching back, but I've had a bunch of sports injuries in my life, and they were much more devastating. I had a bad ankle sprain, multiple ankle sprains, but one of them where I really had no strength in my ankle for a year post, right? And as you said, if I had broken a bone, I would have been back on my feet a lot sooner. Also, to your point about like how it's so mechanical, the orthopedic treatments nowadays. I had a couple shoulder repairs. And I realized how mechanical it was because I ripped out the first repair when I redislocated my shoulder, and to try and get my shoulder back into the socket, I like physically lifted my arm, and I could feel in like throughout my entire body as each like pin that was anchoring the repair was like ding 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 as they shot out. So I was like, wow, I'm really like a old wreck of a car here, and my orthopedist is a mechanic. Um, and that's like not an exaggeration. I mean, a lot of orthopedic medicine is mechanical. And one of the great benefits, I would guess, of cell-based therapies is they provide both like the physical substrate along with biomaterials for repair, as well as that signaling, that the living component that drives and directs regeneration of endogenous like neighboring tissues and integration with those tissues at the site of injury. It's this living implant, right? As opposed to their very elaborate, I don't want to denigrate these surgeons, they're amazing. They have these elaborate repairs, but they're inert for the most part, unless you're talking about allograft, as you alluded to there. But nevertheless, these inert implants are the mainstay of orthopedic surgery in the modern era. And you started that last answer by talking about going to these meetings, right, where you have the kind of cell people, the developmental biologists, and then you have the mechanical, clinicians on the one side. You kind of alluded to it there at the end of your answer with like incorporating biomaterials and cells. But do you envision ever therapies that combine both like that, like inert kind of mainstay approach of modern orthopedics and surgery, but incorporating cells? Is there like a kind of bridge period we're going to have like implants or repairs that are doped with a living cell component, or are we going straight to like a cellular and biomaterials as an alternative to the mechanical approach?
I think the challenge is with cells, right? Cells don't have the kind of mechanical properties, so we cannot expect them to perform the same function as they do when they're embedded in matrix. So to me, there are two options that we don't know which will prevail. So one option that is kind of tried in other tissues is fully engineered tissue, right? So you combine materials with cells outside of the body. You stimulate them. In tendon, there's a lot of stretch that is needed. Biomechanical cues. You form this kind of fibers, right? And then you implant them. So that's probably most predictable, but also difficult to perform strategy. The other way will be to combine materials and cells, materials that already have some close enough biomechanical properties, right? So materials can perform on day zero, but what the cells will do is lay out matrix gradually and gradually replace the materials. So the idea is that the materials will dissolve and disintegrate together with cells laying matrix. So over time, weeks, months, right? You replacing the materials with actual matrix and tissue. So we're looking at both strategies, right? So both have advantages and disadvantages. The advantage of plug and play, right? Tissue is it's very predictable. You know what you're implanting, right? It's the surgeon takes it out, stitches they do allografts or autografts now. So that part will be very simple. The difficult part will be to really engineer these grafts without, vascularization and all that.
Yeah, I think it's an interesting approach. And I think a lot of folks in the stem cell field are trying to figure out the right way to marry the bioengineering and the cell-based approaches. I mean, we've talked to other folks on the show who are doing similar things with muscle and even with cardiac tissue. So I think it's a cool approach. I actually wanted to go back to the back pain question since that came up a lot earlier. And actually, diving into one of your papers, which is specifically focused on this particular topic, you had a paper published in Science Translational Medicine, which is like one of my favorite journals, focusing on back pain. And your lab actually figured out that there's a specialized cell type called the nucleus pulposus cell, generated by certain conditions in degenerating intervertebral discs. And maybe that this is the cell. This is the responsible candidate for the onset of back pain. I mean, along the way, you used a ton of single cell mouse model and even some cool organ on chip models to figure out how these cells might actually contribute to back pain. I'm a fan of organ on chip, as you know, as your next door neighbor here, and everybody at the Regenerative Medicine Institute here seems to be working on organ on chip, right? So tell us a little bit more about this work, maybe how you actually developed that organ on chip model and some of its applications.
Right. So it's funny that this is I think the least translational project we had in the lab, and it got published in Science Translational. But the idea of this study was really to, as we alluded to it earlier, to find out the cause of low back pain. So we know that approximately 40%. Again, that's a figure that everybody throws out, but nobody really knows to quantify that 40% of low back pain is discogenic pain, meaning it originates in the degenerative intervertebral disc. Then that is in healthy condition, is not innervated, not vascularized. So you're not supposed to feel anything that happens there. But in 40% out of 80% of the population, we do feel pain that comes from the disc. So how does it happen? This is question number one. Question number two is if we MRI our discs, all of us, right? Probably most of us have some kind of degeneration of the disc, but not all of us have discogenic pain specifically. So even you, Dalen, you don't know where your pain is originated. It might be from the muscles, it might from the facet joints. There are different reasons that the back pain can come from. But if we look at structure, and that's what we do in the field for many decades, we look at the structure of the disc and say, well, this is degenerated. It's probably the cause, but it's not always, right? We only look at back pain patients. We don't look at patients that are not coming with a back pain issue. But when we do, we know that they're all degenerated. So our specific question was, well, what makes one degenerated disc painful, and right next to it, or a person next to it, this is degenerated, but it's not painful. And that trigger between degeneration and pain is probably not just more degeneration. It's not the more degenerated, the more pain you have. So we really wanted to understand this trigger and what makes some discs be innervated, right? So the sensory neurons are innervating the outer part of the disc, but then we know from sparse kind of histological studies or tracing studies that these neurons innervate the disc, and now we feel pain where we're not supposed to. So in this study, we looked at from, as Arun mentioned, from different angles. We took single cell data from patients and cadaver discs that were degenerated but not painful, and compared, what is different, what makes, one disc population, cell population, inducing pain and one not. And we found this specific cell subtype, right? So a group of cells that express different type of genes and different type of signals that were unique to that or prevalent in that population of intervertebral discs. And then we try to, it's difficult to isolate them, right? So how do you pick them? Surface markers are not great for that. So we try to mimic this population and recreate this population in vitro. We know what are the degenerative conditions in disc. We know the pH is changing. We know that it's overloaded. The vascularization or diffusion of nutrients is compromised, so they have much less glucose around. So we recreated, and there is some inflammation. So we created these conditions in vitro with a small bioreactor, and basically took healthy cells and made them stressed and inflammatory, and then compared their transcriptomics to the ones that we found in actual discs, and the similarity were pretty stunning. So we thought, okay, this is what happens. The cells get stressed in this degenerative environment, and then they start producing these help me, innervate me signals that induce innervation and pain. And we confirmed that by injecting these cells into healthy rat IVD, so not degenerated, and we saw pain outcomes, so without degeneration. So the discs that we injected the human cells were perfectly fine structurally, and there is no inflammation, no degeneration. Only presence of these once stressed human cells caused pain outcomes in those rats. So these cells by themselves could initiate the process that happens in the disc, innervation of the disc, and pain, chronic pain outcomes in those rats. So that was the conclusion, that there is some conditions in the disc that the cells in the back pain population have this trigger or process that they start calling help and induce low back pain.
Yeah, I mean, look, you said this was the least translational story in your lab that went into Science Translational Medicine, but this is translating directly to my life. So I would disagree with you there. And I think the key there is because it's like this whole bundle of cause and effect that's difficult to unravel. And I think this really was instrumental in showing a kind of cell of origin, and therefore kind of mapping out a pathway toward therapeutics. And I mean, I think that's pretty translational. And I was really what I thought was the most impressive. I mean, in spite of like the organ on chip stuff is also amazing toward many purposes and really tractable. But I don't think it displaces the need for, I'm sure you would agree, need for animal models as a really critical medium for the preclinical investigation, interrogation of all these like therapeutic endpoints that incorporate this bundle of cause and effect. And as you said, like it seemed like this cell of origin had a output that was pain sensitivity. And that's the thing, that back pain is kind of amorphous. There's so many contributing cellular component, this biomechanical structural component, that like nerve, electrical impulse signaling component. So you really do need an in vivo platform that can incorporate all of these elements. And to your credit, you've done that, and I think are taking it to the next level. I looked into some of your funding. So you got this big grant as part of the NIH HEAL initiative for analgesic development. So congrats on that. Big funding. And necessarily big, because the proposal puts forth this plan for applying iPS-derived induced notochord cells. Is that out there?
Right.
So these induced, well, presumably to generate these nucleus pulposus cells and to incorporate them in a rat model of this intervertebral disc-degeneration and discogenic back pain. So, I mean, the first thing that jumped out at me is how do you measure the pain reducing capacity in these rats? Like, how do you get that output of pain and quantify it? And is that the primary output, or the cellular outputs for showing regenerative potential of these cells? Also, selfishly, as someone who'd like to see these cells in clinical trials as soon as possible, do you need to go, as this rat model, a sufficient threshold, you think, to move into human trials, or do you need we all walk upright? Is that even relevant? Like having a versus a rat or even a pig? Do you need to have like a large animal or primate trial as a prerequisite to human testing? A lot of questions there, but really that last one is what I'm curious about, because I would like to enroll in that trial, please.
So first, I want to address the organ and shape versus animal model question. So in this study, what we did on organ and shape was to take iPSCs, differentiate them to nociceptors. So we really wanted to see the effect on human nociceptors, because animal models have their advantages, but it's not human. And we always think about, well, it works in the rat, will it work in humans, right? So is that the same mechanism in human cells? And that what organ on chip technology gives you. It only answers very specific questions, right? So we wanted to see our cells, the nucleus pulposus cells that are inducing pain, can they affect specifically nociceptors without all the environment and all the mess and noise, right? So that was really the question, and the answer was that these cells induced ingrowth of axons from one side to another, and that's what we think that happens, right? It's a direct communication between nociceptors and these nucleus pulposus cells. Something that is really difficult to see in animal. You can, stain the discs, you'll see one fiber, maybe, and that's how these conclusions are drawn these days, because it's really small event that it's difficult to see in 3D of the tissue. Another question you asked about how we measure pain, and I think this is really critical, and all our musculoskeletal field slowly moving towards pain and function, and less on structure. We were using primary outcome measures as structure for too long, right? So because it's easier, it's quantitative, it's unbiased, mostly, but it's not the right thing to do. It's not the main outcome. People don't come to the office saying, "Well, my MRI of the disc looks bad. Can you do something about it?" They say, "I have pain," right? So I think moving from primary structural measures to primary pain outcomes and by behavioral tests, that's where we are going, and that's where the field is going. So there are, unfortunately, we cannot ask them to grade their pain in the morning from 1 to 10, but there are indirect measures by behavioral tests that, a battery of tests that we can see. It's noisy, it's messy. You need huge cohorts of animals to kind of sift through this, but this is the main outcome that I think that we should because that's what people care. And in that study that you mentioned by Hill, this is our focus. We create low back pain in animals, and we want to see reduction of that pain without treatment. Whether, yes, we want to see our cells survive, we want to see our cells produce matrix, we want to see what they're doing, but that's a secondary concern, right? So if and that's for any cell therapy in my opinion. If you have your therapy, the cells are gone, you never find them, that's not a failure. As long as the function is restored, as long as the pain goes away, we did our job. I think specifically in the IVD, we should restore this, matrix environment. They have nowhere to go. They're kind of trapped in this tissue. So, and we saw in different studies that they can survive and maintain their phenotype. What we're trying now is to really enhance their ability to secrete matrix and rebuild the tissue in this very challenging conditions of degenerated disc. But the main focus is really to restore the pain outcomes. And to your last question, so I think in many disorders, the transition from small animal to humans is possible when it's very systemic effect that you're looking at, right? In the disc, specifically, size matters. So size of the disc means distance that the nutrition travels, right, from the end plate of the bone of the vertebrate to the disc, and it's very different in rat and humans. And a lot of research been done on mice and rats, and a lot of successful therapies were developed that never made it to clinic because the size, from mouse to rat is 10 times, right? So let's not think about rat to human, right? So what we are doing, we're using a porcine model, so pig model, because the disc is relatively similar in size in humans and pigs, and also you alluded to the upright. That's a comment that comes in every grant, and there is the same answer. So we know that the biomechanical pressure on the disc in pigs, although there is very similar to upright humans, just because of the massive muscles and bones that are pressing on the disc. People actually measure the forces, and they're very similar. So in our model, we degenerate the disc by puncturing it. It's not the same as aging and kind of chronic degeneration. But in a recent study that we're doing right now, we're really trying to characterize this degeneration that happens in pigs, and to see if how similar it is to degenerated disc in humans, and it's quite similar. So we can see the same effect. It's not the same way that this disc degenerates, but the result is very similar. So degenerated disc that was punctured several weeks ago in the pig, and disc that of 60, 70-year-old that slowly degenerated. In the end, they come to the same or very similar place where we can start thinking about regenerating. Another point is for cell therapy, even if we cannot fully regenerate the disc, right? So here is completely destroyed disc, and now if we inject cells, it suddenly becomes 10-year-old. That's probably not going to happen. But that might be an option for stop this process early on with minimally invasive injection rather than full-blown spine surgery. So there are some, ethical and commercial questions probably relevant for any preventive therapy, right? That will be more effective, cheaper to do it earlier, right, than later. So in all medical fields, but I think we should always think about that option as well as society, where we can prevent these things much easier than treat them when they're already, in a severe stage. Did I answer your questions?
No, that was a great answer to Dalon's multi-part question. So, no, very well done. The one thing that I loved, the quote that I loved was that if you can make the pain go away, even if the cells go away, then you've done your job. And I think that is in contrast to a lot of the other cell therapies that we discuss here on the show. For example, neuronal cell therapies, dopaminergic neurons for treating Parkinson's or whatever, where if the cells go away, you've got a problem. So, the end outcome of your studies is to alleviate pain. And, it doesn't matter how you get there. And which is why I think, you're in a great place to do the kind of work that you do, this very translationally oriented work where you can collaborate easily with clinicians who just so happen to be under the same roof where we work, as basic scientists. You're, of course, my partner in crime here at Cedars-Sinai, which I feel like I have to ask you a question about Cedars as our home institution. I hope the higher ups are listening here. You've been here at Cedars for more than a decade, starting as a postdoc before becoming an independent PI. And most recently, thanks to your recent success, like we talked about here on the show, you've been promoted to associate professor. So congrats again on that. Cedars, as you know, is kind of an outlier among traditional academic institutions, and that, like what we're talking about, the basic science that's being done here is almost exclusively translational. And I think a big part of that is, of course, we're a hospital first and foremost. We work literally like under the same roof as the clinicians. You can go down 2 floors and go to the clinical departments, right? I mean, we get to walk by the patients who are, in your example, experiencing pain every single day. We walk by them in the lobbies on our way to work. It really gives you a nice perspective on the importance of our work as basic scientists. So for you, what's been so special about being here at Cedars that's really uniquely enabled the work that you do?
So great points. Yes, orthopedics has a department, has tens of surgeons that are not only in spine, right? We collaborate with different subdivisions in orthopedics, and yes, the fact that we're on 8th floor, the ORs here on the 5th floor, right? We 3 floors down, and we get access to surgical samples, like what I was mentioning for the back pain. We also do some work with modeling OA, and there are tens of patients going through surgeries each week, and access to those tissues is huge advantage, and access to clinicians that are hungry for those therapies is an advantage. So besides that, Cedar has a lot of vision towards translation and biomanufacturing. So that's the next step. Once we figure out how to make these cells in a lab, the goal is to manufacture them in scalable and transportable quantities that will make this therapies available for not only our patients, but patients in the periphery, right? So, for example, and part of the HEAL grant is to see how can we transport these cells. Actually, one of our aims is to, can we do we have an incubator to transport these cells? Can we freeze them? Can we ship them on ice? Because that's the injectability of this therapy makes it available for not only big academic centers, but in small clinics as well, right? So that's the goal, to manufacture injectable therapies, put them in a vial, and ship it overnight to anywhere in the country, right? So that's also a part of the vision. And as you mentioned, CIRM, California Institute for Regenerative Medicine, is also very supportive of that work. So we have a grant on the porcine model, right, to develop that in the pig. And if we see that this is working in the pig, that's a huge promise for human trials as well.
Yeah, I mean, I love New York, but I am a bit jealous of the situation you guys got going out there at Cedars. And because, I mean, it is Cedars itself, right? As you described, between the patients and the, the investigators and the physicians and surgeons, and the funding, right? The resources from CERN, but also from Cedars, let's be honest, it's a very, rich hospital, rich clientele, and they pay that a lot of that revenue forward to the research, not just for the good of mankind, but that's good for business, right? Developing the next therapies. And kind of to that point, the culture, I think more than anything, in a major academic center, but in California, where there's a really strong legacy of innovation across all fronts. I mean, you guys have it. You've got it all. And I love to hear how you're talking about like the real practical measures you got to take to move these therapies forward. That's what's so exciting about the time we live in, is that like the proof of principle in large part has been demonstrated. And now it's about scale, it's about cost, it's about, making these things available in a way that's practical and efficient. And it gives me hope that I can put aside my anguish at the unpredictability and vicissitudes of my chronic pain. But I'm sure I'll have to wait just a little bit longer, but hopefully not too long, the way you're cooking over there, Dmitry. Before we let you go, we got a couple of science adjacent questions for you. First, what's the biggest misconception about science that you would like to resolve?
So my answer is more about stem cell science. Okay, I think we kind of alluded to it a little bit, but we have this, and not all of us, but some people have this misconception that the only way to regenerate tissues is to replicate development, and I think it's impossible, right? So development is a kind of 4D process that is not going to happen in injured or aging person, and by we can mimic development a little bit. We can hijack some mechanisms and maybe piggyback right on these mechanisms to when we're trying to regenerate tissues. But if we can find some shortcuts, right, and get to the point where the cells and the tissue is functional, although it's not 100% original, right? And again, like in a 10-year-old, I think we'll be fine. So it's more about function of the tissue rather than the process, how we get to that. So that's I think we have to think about the end product more than the process. Although we enjoy learning about that through development and using that knowledge for differentiation, it's never going to be the same. And the most important part of engineered tissues and the most valuable feature is that they can remodel and adjust to the patient, right? So if this tissue can integrate and then go through the modeling process, then it will be natural and already fully integrated, unlike all the hardware that cannot remodel. So if the tissue is remodeled, we're fine.
Yeah, again, that circles back to the kind of idea that Arun echoed, that, if the cells are there or not, it doesn't matter if we reach our endpoint, which is getting the job done. And like stem cells, let's be frank, it's a big hack. Their existence is really transient in development, and their sustained culture in vitro is a kind of an artifact of our will.
Yeah.
Right. And similarly, from a pragmatic standpoint, whatever we can do to get cells that get us there therapeutically, I'm just fine with that. Finally, we have a few fill in the blanks for you. First, when I'm not conducting research, I am
Driving my boys to practices and games. That's probably most common. But that if not, then I'm probably gardening while listening to audiobooks.
Yeah, I've been there. The drive, and it seems like you put in like 10, 20 hours a week just as a Uber driver for these kids. But it's a great joy of mine. I'm sure it's the same for you. Next, if I could have one superpower, it would be.
So I would like to inspire people and to be motivated and curious and excited. I'm trying it every day, but I think it's. I would wish to have a superpower to do it more efficiently. Um, I think that's the my real wish.
Well, somebody get this man a cape, because you've inspired me just in the scope of this interview, and I'm sure I'm not the only one. Finally, I can't start the day without
Cup of coffee or two.
Yes, keep it simple.
Yeah,
I mean,
yeah,
got to get the fuel in, right?
yeah. It's my favorite addiction.
Dmitry, man, thank you so much for joining us today on the show. I'm not just saying it. This was an inspiration for me, because this is a thing when you talk about something where you have a personal, it has a role in your own life. It really gives you a different perspective. So thanks for your work, and thanks for sharing all that you do and talking about it with us.
Excellent. I enjoyed that conversation too.
All right, everybody, that brings us to the end of our show for today. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, or by email at info@stemcellpodcast.com with feedback or to suggest guests.